Alright, Assalamualaikum warahmatullahi wabarakatuh. Very good morning. I hope nobody actually went to KDK because WhatsApp, nobody replied. So I assume that nobody's coming. Okay, so what we have today is we just have an online class. Alright, so there's only 20 of you here. Okay, never mind. Just expect that every one of you are actually doing your assignment, so I'm okay. Submission is next week, right? So after this lecture, I will open a link on Spectrum for your submission in case if you're already completed and you want to submit, then you can. Okay. Since next week is the last week for me with you guys, looking at this particular course, so I just want to go back and look at a little bit on the learning objective for the program, for the course, not program. Okay, so basically, number one, the course learning outcome is to recognize basic principles involved in drug research. We did have cover about this a while back in the first two lectures. So, you can just go and refer it back. Okay. So what is the important thing about CLO? If you don't know, okay, all your exam questions will be based on the respective CLO. Okay, so in this case, this course has 3 CLOs. So definitely your exam question will cover all 3 principles. Okay, so first one, recognize basic principles involved in drug research. So definitely you need to understand how drugs are being developed in general, obviously. Okay, you don't need to know like in very fine detail, but yeah, generally you need to know what are the pathway. And then the second one is for you to describe types of drug, mechanism of action, structure, activity, relationship, strategies for drug design and development, and okay, so that's number 2. So you can also expect questions will come out because we've looked at multiple drugs. So there will be some questions which relates to types of drug, mechanism of action of those drugs, or all the drugs that we've covered. And of course, most particularly important is the structure activity relationship. Okay, so this is also part of what you are doing for your assignment. Okay, so you'll be focusing more on CLO number 2, a little bit on CLO number 1, because what you are doing is for the assignment is generally how the modern drug discovery are being developed. Okay, so basic principles in drug research involve the design of the drug. Therefore, what you are doing now is pretty much on the first step of the drug design. Okay, and then number 3, which will be covered by Dr. Diana, is basically on the pharmacokinetics of drug metabolism. So normally, if you read research articles or even textbooks about drug research or drug discovery, they always combine this keyword pharmacokinetics with the other one, which is pharmacodynamics. Okay, I've covered I think 1 or 2 slides about pharmacodynamics, but that is something again you just need to know, but you don't need to know in very fine detail, like what experiments you can do and stuff like that. Okay. All right, and the program outcome for this course again is PLO1, which is master knowledge. What does it mean by master knowledge? Is basically for you to memorize the content. Okay, so so this one is more on memorizing, number 1, because it's very straightforward, and PLO6, which is a more of a higher level thinking. Okay, where you need to identify, analyze, and solving problems. Okay, so it won't be it can be as straightforward, but mostly it will go under more on you need to identify, you need to memorize, and solve a problem. Okay, and I've completed all the lecture notes, and I think we can do a tutorial on the 7th on our last lecture. Okay, so next Thursday, then you probably see a few types of these type of questions. Okay. All right, so now we are on this day. We're going to cover about Analgesic part 2, and then part 3 we will cover on next week. Okay, and hopefully we can do that on tutorial, or perhaps a little bit, probably about 20 minutes or so to cover all the remaining lecture notes, and then for the rest of it is more on Pol F, so that you can try and see a little bit of type of questions that can be asked. Okay. So we've looked at anesthetics and analgesics, about the history last time, a bit of the classification, but we stopped somewhere there. So what we're going to do today is going to continue on the classification. Okay, and first we've also looked at pain. What is pain? So basically, it's just a signal transduction from the nociceptors on the skin or on the epithelial cells. That's a more proper word. Okay, epithelial cells, basically all the cells covering your body in and out, meaning that you have receptors on your skin, but at the same time, your mouth is also kind of like, you do call it as a skin, but you do have a layer of cells within your mouth, oral area. Okay, so these are all epithelial cells, and epithelial cells, they do have nerves, and they do have nociceptors. Okay, and also we've looked at two types of complex systems by which pain can be perceived. Okay, so we do have two receptors, part of the system, which is cannabinoids, and second one is opioids. Okay, cannabinoids, it's pretty much just a receptor that, well, it is called as cannabinoids because originally it was discovered to be the receptor that is responsible for cannabis molecule binding. Okay, so that's why we call it cannabinoids. And opioids, again, because opioids is like morphines and whatnot, are the drugs that was used originally before the receptor is even perceived, even discovered. And then once it was discovered, the receptor binds, or opioids binds strongly to this receptor. Thus, the receptor is called as opioid receptors. Okay, so that's the naming. Looking on a little bit of detail, opioid receptors, there are three main receptors that are currently being discovered. So there are a fourth one, but I'm not including that as part of the learning process. Again, this is just for you to know. It doesn't mean that you need to really know what are the differences between these three. Okay, so we have mu receptor, kappa receptor, and delta receptor. Okay, and opioids, both opioids and cannabinoids can be found in more abundance on nerve cells. Okay, but opioids are mostly on the CNS, the central nervous system. Okay, it doesn't mean that it's not there in the peripheral. It is still there, but it's mostly concentrated on the CNS. Okay, so CNS contains, again, this is, if I'm mistaken, just high school. So the central nervous system includes the brain and the spinal cord. Okay, of course, brainstem also is part of it. It's part of a brain. While the peripheral system is more on the ganglion, the nerve cell branching out from the spinal cord. Okay, and the other nerve that kind of like, is branching out from the central ones. Okay, so you look at the functions, general functions about both opioid receptors and cannabinoid receptors. We have looked at CD1 receptor in the nerve structure last time. I'm going to re-show it to you guys after this. And for the opioid receptors, again, we do not look into fine detail where is mu, where is delta, where is kappa. But suffice to say that, it's a receptor, there's different types. And so if you are planning to target certain things, for example, if you can see here, each receptor has its own specific function. There are some general ones that cover both or even all three of the types of receptors. Okay, for example, you can see here sedation over there, analgesia, spinal analgesia, it's there on mu, delta, and kappa. Okay, so There are some that are similar in all three, but there are also which are a bit more specific. For example, gastrointestinal, there's a kappa opioid receptors, but there's lack of, it doesn't mean there's no, but there's lack of mu and delta. Okay, towards the other day, for biology, you can't really say there's nothing at all, because there could be the receptors in some other person, not the person that they are investigating. Okay, so generally these are the distribution, um, and we do have endogenous, what we call as endogenous opioid peptide affinity. For those who might be confused, what does it mean by endogenous opioid peptide affinity? It means that endogenous, well, you learn about endo exo, right? So endo is something that you keep inside, exo is something that you from the outside. Okay, either you put it out or something like that. So that's how I remember. Endo, it's something to do with inside, exo, something to do with outside. So what it means by endogenous opioids peptide is that the peptides are actually inside our body. So it's not something that you consume, like for example, if you're taking opioids drugs or morphine, for example. So morphine is an example of exogenous, okay, because it's external and then you want to take it in. But these endogenous are basically peptides that your body actually produce. And of course, if there's a receptor, then your body definitely will use that receptor for a certain function. Okay, so that's when we do have this endogenous opioid peptides. Okay, so an example is endorphins, enkephalins, dynorphins. Okay, and you can see they are more or less the same. Okay, but you can see the distribution is slightly different. Okay, dynorphins are more preferred. Okay, the double arrow means it's more preferred in the kappa receptor in comparison to delta or mu. Okay, so that's the differences. What it also means is that if you have dynorphins, okay, and then you eat it, compared to if you inject it. Okay, so if you eat it, because dynorphins are present or in more abundance in gastrointestinal tract in comparison to the other sites, then you can expect that the influence of that particular drug are higher in the gastrointestinal compared to in the other side of the system. Okay, so that's what it means. CB receptors, on the other hand, the cannabinoid receptors involved in motor activity, thinking, motor coordination, appetite, short-term memory, pain receptors, immune cells. Okay, so towards the end of the day, part of it controls the pain. Okay, so that's our goal for today. We're looking at pain. Okay, while CB2, you have all the other things. There's CNS, even though it doesn't really mention about pain, but because there's involved CNS and you do know that the receptors are highly abundant in CNS, then CB2 can also take part of pain control. Okay, but again, biology is very complex. As you saw previously in this slide, you have so many receptors, even though if you consider the number of neurons are only 3, okay, the blue, green, and red neurons, but there are so many receptors, there's so many transport proteins in each neurons that have a certain function. Okay, so we have CB1 over here, which is the cannabinoid receptor. The opioid receptors are not shown here. So what I did, okay, and another one more thing that you need to kind of like try and remember, okay, for the next, until the next week, is this, okay, AA. We're gonna touch on AA most likely next Tuesday. Okay, so what is AA? AA is, where was it? That, okay, is arachidonic acid. Okay, so we will see the structure about AA and how AA actually involved in pain perceptive. Okay, so this is also this is just another slide showing you guys on the opioid receptors, and if you actually read on the right hand side, it's pretty much, it's very clear cut. it's actually a very good representation on how pains are being transferred and how opioids actually relieve pain, so to say. Okay, so we've looked at how pain transfers. So you do have your basal, so action potential. Right, just again a recap: action potential is when the signal is being transferred through the neurons. Okay, so in the normal scenario on the left hand side, where you have an action potential, when it reaches on the dendrites, so what will happen is you the information that is being carried, or the potential that is being carried, pretty much activates calcium channel influx into cells, CEMP, cyclic EMP, production and synthesis, and therefore release of neurotransmitter. Okay, so it releases neurotransmitter from one neuron transferring to another neuron. Okay, and once it reaches the other neuron, keeps on transferring until it reaches the brain, then that's when you feel pain. Okay. However, in the presence of opioids, what will happen is if you have opioids will activate opioid receptors, and what it does is pretty much it inhibits these two other transport proteins, and because there's no because the transport protein is not being inhibited, that then it cannot do its normal function, which is towards the end of the day releasing the neurotransmitter. So if you see here, opioids blocks neurotransmitter, so that's one path. Okay, so because the signal can now not be transferred from your nociceptors or your pain receptors to your brain, therefore you will not feel pain. It doesn't mean that the signal is not there. The signal is still there. Your nociceptors keeps on trying to send the signals. However, due to the blockade between the synapse or the junction between two neurons, thus your brain do not process what pain is, and thus you don't really feel pain. Okay. At the same time, the opioid receptors on the receiving end of the neuron, so the other end, cause hyperpolarization. Okay. So why is it important? Again, if you recall on your high school biology, nerve send signals via electrical impulse, right? So you have kind of like a threshold, and then once the stimuli reaches the threshold, the signal will depolarize. Okay, and then after that, it will be on the recovery period. Okay, so that's the part of a nerve, how the how do I call this, the current in the nerve is seen or is perceived. Okay, so this is when you have the action potential. Now, what hyperpolarization means is that it lowers the signal. Okay, so instead of it activates, so it lowers the signal even further. So these are the because this one is normal. Okay, at a resting stage, this is how your the current value, not current meaning sekarang, but current electricity. Okay, the current value in millivolt of our neurons are, so that's the normal baseline. But then when you have this hyperpolarization, the signal actually deepens, it gets even lower. Okay, so if the signal gets even lower, then definitely there will not be any signal, and no signal meaning no information being transferred. Okay, so you do have some other different types of mechanism. You can just go and have a read. Okay, and also read on number 3, C, on how the signal is being transferred and how opioids are actually stopping the signal from being transferred. Okay. Over here, the important things are, well, generally on opioid receptors, there's no specific on whether it's mu, kappa, or delta. That is something for you to know. It doesn't mean that you need to memorize, oh, delta is doing this, kappa is doing that. No. Okay, so it's enough for you to understand a little bit. So the take point is again, there are multiple receptors, and these receptors are normally as G protein coupled receptor or GPCR receptors, and these GPCR receptors can either activate, okay, being activated, so not they are activated, being activated or deactivated to control pain. Okay, so activated means that there's an agonist, deactivate means there is an antagonist. Okay, so there's a molecule that either activates or deactivates the receptor itself. So we'll look at a little bit, very briefly, I think next week. Okay. So again, cascade of signals via two complex receptor systems. We do have cannabinoids and opioids receptors, as shown in the previous two slides. Okay. And the other ones are not really specifically on either opioids or cannabinoids, but it's related. Okay, so we haven't touched about analgesic base that does not really go into steroids. Okay, so opioids and cannabis, we call it as a steroid. So these are all steroidic receptors. Okay. Okay, steroidic receptors. So we do have another side of this coin, which what we call as the non-steroidal ID. What's the full name? Non-steroid anti-inflammatory drugs. Okay, so we don't call it NITES-R, but it's kind of like the other drugs that are non-steroid. So because they are not part of a steroid, which is cannabis and opioids, then you can expect the mode of action are different. Okay, so NSAIDs and the steroids or opioids or cannabis act in a different manner, but towards the end of the day, both can somehow relieve pain. So we will look at this one next week. Right, so both receptors coupled to G protein and their activation leads to inhibition of adenylyl cyclase and potassium channel activity, ultimately leading to inhibition of synaptic transmission. Okay, so you inhibit, you block that, so there's no signal, so there's no pain. So no signal, no pain. Okay, so take home message, there are multiple receptors. Again, probably I just forgot to erase the previous slide. So this one should be the summary for the previous ones. Okay, so we are not going to look at cannabinoid receptors, but we're going to look at opioid receptors and opioid peptides, the endogenous peptides that I mentioned. Why? Well, just because we don't have enough time to actually cover both. Well, pretty much the principle are the same, but opioid peptides are, for me, more interesting because you know, we do have, well, a long time ago, not too long ago, we do have issues with morphines, for example, morphine addiction and whatnot. So it will be best to look into this rather than cannabis, because cannabis, still interesting story, because you know how cannabis is legalized, is legal in US but not legal in Malaysia. But you know, that's a different story. Probably your junior, I will talk about that instead of opioids. Now, there are generally five types, not examples. Okay, five types of opioid peptides that have an effects on either mu, delta, or kappa receptors of the opioid receptors. And you can see here all of these. Okay, you can see a single star and a double star. A single star means it's an endogenous peptide. Again, I mentioned it's a peptide that is being produced by our own body. Okay, so we produce this, even though it's acting on opioid receptors. It doesn't mean that we are trying to get ourselves addicted, but we'll talk about that in a few seconds. Okay, and the double star means exogenous. So if endogenous, again from inside, exogenous is from outside. Okay, like morphine, heroin, and whatnot. So they belong to the same category, and therefore, but I'll just put that as one example. And you can see here endomorphin 1 and 2. So that is the peptide sequence. Okay, endomorphin 1 and 2. act primarily on the mu receptor. Again, doesn't mean that there's none on delta and kappa. It's probably just mean that it is still not discovered. Okay, so biology keeps on evolving. We kind of like discovering more and more stuff. But yeah, and enkephalin, for example, that's the sequence. Is more active on delta compared to mu and kappa. Endorphins, which is primarily the pain relief, okay, is acting more on mu and delta in about equal manner, not so much on kappa. Dynorphins, as we recall, it is acting on kappa, and at the same time, it's more on the gastrointestinal tract. Okay, so it's more kind of like a bit more specific. And morphines, it's more effective in the mu receptor. So if it's kind of like active in the mu receptor, then you can expect that morphine can also become like a competitive inhibitor, or it can actually activate the receptors in these two other regions, okay, or the effects of these two other molecules. Okay, so again, endorphins are pretty much pain relief. Okay, however, it's not just that what it does. So endorphin are being released by our brain, pituitary gland. If you recall, we have a brain kind of like that, and then you do have like a peanut shaped thing in the middle of your brain, and this is where the endorphins are being synthesized and being released. Okay, so you actually get endorphins from exercise, doing acupuncture, meditation, sex, listening to music, laughter, UV exposure. So it's kind of like when we're talking about pain relief, it's something that makes you kind of like happy, right? So that's the opposite. So either you be happy or you feel pain. So pain or stress, it's kind of like a bit difficult to justify what it is, but nonetheless they belong to the same group. One is negative, the other one is positive. Okay, so that's why some people like to listen to music, some others don't. Some people like to laugh, some, well, that's why if you smile, it's kind of like a bit happy, not so much about happy, but a little bit happy. So you are actually releasing a bit of endorphins, and thus you don't really feel pain and feel more kind of like relaxed. Okay, there are more than 20 times natural isomer for endorphins. Okay, and yeah, as I mentioned, it's kind of like related to pleasure sense, feeling of happiness. So what we will do next is we're going to look at pain relief via anesthetics and analgesic on opioid receptors, and we're going to specifically look at morphine. Okay, so that is what we just saw just now. Okay, morphine binds strongly to mu receptor. It is an agonist, so it activates the receptor. So again, mu receptors, if you're talking about endorphins just now, endorphins are related to the feeling of pleasure. So that's why if I'm not sure if you have, but if you have been either in a surgical, you've been into surgery, a major surgery, for example, normally you'll be given morphine, okay, as a pain control. And if you actually see the person who are actually under morphine, kind of like to laugh, smile a lot, so it is actually because morphine is actually activating a mu receptor in the same way as how endorphins, the happy molecule, activates the receptor. Okay, so that's why the kind of like the effect is more or less the same. You can see both are the same, but the difference is endorphins in I can't remember, if I'm not mistaken, is in the microgram concentration, can have like how we are happy. Okay, so the happiness, the natural happiness or pleasure that you can actually feel. But morphine, on the other hand, if you actually take a very tiny bit, you can be happy in the same way as how endorphins makes you happy. But the difference is in terms of drug abuse, when people actually take in morphines, they take in a high concentration. So that's why the feel are different. And because morphine is something that you can control, imagine if you can actually control endorphins, probably you get yourself addicted very, very easily. You know, you can just laugh and then you feel happy. Probably you can just keep on laughing forever, and then until you die. So similarly, if you inject a little bit of morphine, you feel like, oh, maybe it's nice. So what your body tend to do is you tend to develop addiction. Okay, so when you develop addiction, what will happen is you want to keep on adding more and more morphine into your body, and thus towards the end of the day, at one point you get overdose and die. Okay, so looking at the structure, that is the structure of one of the natural endorphins. Okay, and on the right hand side is the structure of morphine. So just by looking at the space filling model, or this one is ball and stick model for morphine and endorphin, you can actually see there are similarities, basic similarities, straightforward similarities. And from here, you can perhaps predict, oh, okay, so in that case, perhaps this region that has similarities, okay, are the ones that are more important for that particular structure to activate the opioid receptors. Okay, and that is also true, as we will see on structural activity relationship after this. Okay, so endorphin receptors are part of opioid receptors. The naming system is kind of like redundant, because originally it was found to be, opioids binds to it, so you call it opioid receptors. But then another person found that, oh, actually our natural, so like opioids, or probably they don't call it opioids at that time, you call it natural endorphins, actually bound to the same receptor. So we're gonna call it endorphins receptors. Okay. The other person, one group, call it as opioids because they are investigating about opioids. Another one call it endorphins because they are investigating endorphins. But nonetheless, they are both similar. It's just that sometimes naming kind of like make it you confused a little bit. Okay, so on the right hand side is the structure of morphine. So this is how morphine looks like, chemical structurally. And how do you actually number the morphine is by going on the outer ring first, and then you go into the inner ring. So from here, they started off with the phenyl ring over here, because again, if you look at that particular structure on the left hand side, that is actually the structure of a phenol, right? So you have 6 carbons, and then one, the red one is oxygen, this one is an oxygen, and then that one is hydrogen. Okay, so that structure is a structure of a phenol. So perhaps that's why, I'm not sure, perhaps that's why they started number 1 on the phenol. Okay, it's only logical, but I'm not really quite sure because this structure has been solved in from second 18 something. It's a while back, more than 100 years ago. Okay, so anyhow, we start with the numbering on number 1 over here, number 2, number 3, number 4, and instead of going inside the phenyl ring straight away, you jump around to look at the outer ring of the whole structure. Okay, so number 5 is actually over here, 6, 7, 8, and 9 over there. You have 10, 11, 12, 13, 14 for that one. And then for some reason, instead of 15 being here, they number it 15 to be over there and 16 over here. Don't ask me why. I tried to dig for that particular information, but unfortunately I cannot find it. Because it's a bit weird because you have 15 over here, 16 over there, and then suddenly they call 17 for the nitrogen. Weird, but again, it's a very ancient molecule. Perhaps since someone did it, so everybody follows. Okay, so that is the numbering for the morphine. So that there's 2 hydroxyl, for example. So if you need to kind of like trying to refer to 1 hydroxyl, there's 2 ways of doing that. One, you can say the hydroxyl on the phenol, or the second one is you can say the 3-OH or 3-hydroxyl group. So definitely people will know that 3-hydroxyl refers to this particular hydroxyl only. Okay. And then you can also see here there are multiple stereocenters. Okay, so there's 5 stereocenters specifically. So you have 5R, 6S, 4, and then 9R, and then 10R and 14S. Okay, so these are the stereochemistry of a morphine. Now, let's look at the structure activity relationship. And because the phenolic structure is has a similar structure to endorphins, so I started off with looking at how important is this phenolic structure. Okay, so the substitution on the 3-hydroxyl or phenolic, which is that one over there. Okay, so if you methylate it, so we have OME over here, that's when you produce codeine. Okay, codeine, if you recall codeine, we've talked about codeine last time. So originally, a long time ago, it was in cough syrup. Okay? Even before the discovery of morphine, it was codeine. Okay, so and it was used in cough syrup. So you can imagine a long time ago, I'm guessing a lot of people have been, is actually being addicted to cough syrup. And when I was a kid, codeine is still actually being used in cough syrup. So if I'm not sure whether in your generation it is still an issue or not, but a long time ago, instead of how do I say this, a phenomena where people actually smell gum, a glue. Okay, so if you remember a long time ago, or perhaps when you were a kid, there is an issue with hidu gum. So there's one symptoms called hidu gum. But before hidu gum or glue smelling become a thing, a long time ago there was a kind of like similar incident where they actually smell cough syrup. Okay, because cough syrup contains codeine, so smelling it kind of like activates and gets you high. Anyhow, let's go back to SAR. Okay, so methylated, it's improved bioavailability, meaning that there will be more of this particular structure in your brain. Okay, so when you're talking about bioavailability for morphine, is specifically looking at central nervous system, so brain and your stem cell, not stem cell, stem, brain stem. However, it decreases activity. So the activity here refers to how this drug activates the either endorphin receptors or opioid receptors or mu receptor, whatever you want to call it. Okay, so in terms of activating the receptor, if you methylate it, it becomes codeine, no longer a morphine, and thus the activity reduces a little bit. Okay, why? Is because you actually kind of like mask the phenolic group. Again, move back to how endorphin structure is. So endorphin does not have any methylation on the phenolic group, and therefore, if you actually methylate the same group, then you can see the activity will be slightly different. Okay, so you mask the group. However, it crosses the blood-brain membrane, blood-brain barrier better. Okay, so if you don't remember what is BBB or blood-brain barrier, it's generally kind of like, so our brain has its own ecosystem. Okay, so to say. So when you're talking about how our blood travels throughout our body, so it flows throughout our body, like literally flows throughout our body. So the blood from your left hand finger can actually travel. There is a probability, at least, for it to actually travel and then go to your right hand side. So it goes throughout your system. However, for our brain, of course, you probably know that our brain doesn't have any red blood cells, so it's mostly on plasma. And the layer that kind of like separates between the other system and our brain is what we call as a blood-brain barrier. So that's kind of like a layer separating. So you have this is kind of a specific cell that we call as the blood-brain barrier. Okay, and then these are all the other body. Okay, so your other bodies. So that's kind of like a separate. This is actually similar to how a fetus or a baby in a mother's womb. Okay, so the baby and the mother doesn't share same blood. Okay, so the red blood cells of the mom is the mom's, the red blood cells of the baby is the baby's. So they don't share the blood because there is actually a barrier. So the placenta is the barrier between the baby and the mother. So similarly, we actually have our own barrier. We have two barriers generally. So one is we call as blood brain barrier. The other one is what we call as air blood barrier. Okay, so air means lungs. When we are breathing, okay, we are taking in the air from our surrounding into our lungs. But then how does the air actually get into the blood? So there is a barrier that actually helps in the process, in the transfer of the air from the air or the oxygen from the air into our blood. Okay, so AC must be hydrolyzed for the drug to be active. Okay, again, that's what we've talked about. Second one, if you so the 6-hydroxy is this one over there, okay, based on the numbering again. So if you methylate this particular hydroxyl, so you will form a structure as what we call as heterocodene. Okay, it's kind of like codeine because codeine has a methyl group on the hydroxyl. So if you switch the methylation from the 3-hydroxyl to 6-hydroxyl, you call it heterocodene. Okay, simple naming system, I guess. So what will happen is that you actually mask the polar group, and if you mask the polar group, you will increase the bioavailability. So it will stay in the body longer in comparison to if it's more polar. So something that is more polar normally can be excreted out from your body very, very easily compared to a non-polar molecule. So if you have a very, very polar molecule, you want it to stay longer in your body, the easiest way for you to do is you methylate it. So you reduce the polarity. Okay, so increase the bioavailability, and therefore it will cross blood-brain barrier easier, and therefore because your phenol are still as a phenol, it's not masked in any way. Thus, research has shown that heterocodone actually has 5 times higher activity than morphine. Okay, because you mask it, you increase the bioavailability, it gets into your brain even better, but at the same time you have the phenolic compound that is the activator at the active side of the receptor that can do its thing. Okay, so generally speaking, you mask that section, you increase the concentration of the drug inside your brain, and therefore you get increase in activity. So that's basically how the flow is. Okay, and then next one, you have acetylated. So the first one is just methylated, so you form a methyl. Second one, if you acetylate that one, okay, so you have a C double bond O over there. So what will happen in similar manner as how if you just methylate it, you mask the polar group. Again, you increase the bioavailability. It passes through blood-brain barrier better, and therefore again the phenol are free to interact with the receptor even better, and thus you get an increase in activity. But this time around it's all 5. The data shows it's about 4 times greater. Okay, however, it has a very weird, even weirder side effects, which is dependency. So this is where, if you were to compare with morphine, so morphine, say if addiction to morphine is level 1, okay, so this drug, it has more activity, and also it increases the dependency or addiction level to number 2 or number 3. Okay, so it was reported that the addiction is very high, and therefore it is actually even banned in hospitals. Like morphine, yes, there can be addiction, but it is still legal. So hospitals are still using morphines, even though nowadays there's neuromorphines and whatnot, different types of morphine that is less addictive. But nonetheless, morphine are still used. But the 6-acetylmorphine are actually being banned altogether, primarily due to this side effect. All right, and then next one, we're going to look at substitution on number 6 and number 14 hydroxyl. So not just one, we're going to look at 4. So again, 14 just now was 9, 10, 11, 12, 13, 14. So that one over there. Okay, so you can get hydroxyl on number 14 if you do kind of like some chemical reaction. Okay, so why someone discovered that in one step reaction using peroxide, you can actually get an hydroxyl on number 6. Okay, 6 here is 1, 2, 3, 4, 5, and 6. You can get 6-oxo. So instead of a hydroxyl, it now becomes a ketone. Okay, and then you get a 14 on the 14th residue over here. You get a hydroxyl. Okay, so this one's step reaction produces 2 different functional groups, or creates 2 functional groups, and then from here you can call it 6-oxo morphone. Okay, no longer morphine, now becomes a morphone because it's a ketone. It reduces the polarity. Okay, again, because you are converting hydroxyls to a ketone, even though you say, oh, but you created another hydroxyl group here. I'm not sure how they actually measure it, but pretty much they can measure it, and thus they can make a conclusion that it's actually reduces the polarity. So again, similar to the other stuff, if you reduce the polarity, you increase the bioavailability, and it passes through the blood-brain barrier even easier, and therefore you can increase the overall activity. Okay, so that's the simple case for this particular structure. Okay, and then we do have double bonds. We have 2 different types of double bonds. One is aromatic, and then we have another one over here that is on the 7 and 8 carbons. Okay, we're going to look at the aromatic first. So on the C aromatic, if you reduce it, it will reduce the activity. Straightforward. So that's the name of the morphine. If you reduce the that one, pretty much is because if you recall the structure of a phenol structure is kind of like very flat, very straight, it's very rigid. Once you reduce it, there's no longer a double bond over there, then the structure becomes like any other cyclohexane, where it's kind of like fluctuates both and chair conformation and whatnot. So it doesn't really bind as strongly to the endorphin receptor as a morphine. Okay, and then if you remove it, so first one is if you reduce it. Okay, so you reduce it, so no longer has double bond. But if you remove it altogether, it doesn't mean that you reduce the whole thing. You just remove this part, because you still need so just these 2 carbons pretty much. You remove the carbons, and then you remove the double bond as well. What will happen? You will also reduce the activity. Okay, so hydroxymethylazoquinoline. So that's the name of the structure. It's no longer morphine. And then what else? Okay, and then on the carbon 7 and 8, so if you reduce it again to just simply becomes a cyclohexane-like structure, somehow it becomes slightly more potent. Okay, very interesting, but well, nonetheless, it's not my experiment, so to say. Okay, and then what other things that you can change? You can change the nitrogen 17. You can try and play around with the stereochemistry, and then you do have the ether bridge, that one over there. Okay, so on the nitrogen side, it's very important for activity, because on the binding side itself, the lone pair of the nitrogen actually forms hydrogen bond with another peptides on the receptor. Okay, so that's why it says it's very important for activity. If you reduce it, if you change it to something else, then the activity will be lost. The methylation itself, it is important for blood-brain barrier transport, meaning that if you change it to a more alkyl, then you will increase because you kind of like make it less polar. So less polar, again, what we've seen previously, less polar, increased bioavailability, and then it crosses blood-brain barrier easier. Okay, so resulted in agonists, but if you some types you can actually, because morphine is an agonist, so some R groups will pretty much becomes an antagonist itself instead of activate, so it deactivates. Okay, and then if you change it to butyl, you have zero activity, so there's total loss of activity. I have no idea why. It will be very interesting to look into, and that's not the scope of this lecture anyhow. And then if you change it to a structure like this, so you have a two alkyl and then towards the end of the day you have a phenyl group, the activity becomes way higher. So it's very interesting why that happens. Okay, and then stereochemistry is very important for activity. You change any of the stereochemistry from R to S, total loss. So there won't be any activity altogether. So from there, you can kind of like say that the endorphins receptor has a certain shape. So once you change the stereochemistry, you change the structure of the morphine a little bit, and thus it will not bind on the receptor as how it should be binding. Okay. And the last one, the ether bridge is not critical for the activity. However, there is reports of increased potency if you actually remove it. So again, it's there for some reason, but if you remove it, there will be a bit more flexibility on the other chain, and perhaps that's why the activity increases. Okay, so that is just a summary of what we've talked about. You can just read it at your own time. We will stop here and we will continue next week. Okay, so this one is kind of like a summary. Just go and have a read. Have a read. But this is the one side of the coin, opioids. So while morphine can also bind to nociceptor receptors. Additional SAR, we're not going to cover about that, at least for this particular lecture. Okay, so next week, I actually plan to talk about a little bit on your assignment. Okay, so I do have received a lot of questions from your friends asking me, how do I want it to be? So pretty much, it's kind of like in similar manner as how I just presented, okay, especially in the morphine. So that's why this time around, when I'm doing the morphine, kind of like give you like stepwise, right, one by one. So that's pretty much how I would like to read the assignments. I don't want it to be, so at least it's more kind of like arranged manner instead of just haywire, you just jumble up everything. Okay, but not exactly as I presented, in the sense that what presented was an experimental results. So what you guys are doing, well, it is actually still an experimental results, but it's based on theoretical experimental results. It's not experimental looking at the biology of the whole thing. Okay, so just do your best in writing your assignments. Work as a team, do not work individually, and of course help one another. Okay, so that's all from me. That's are the references, and see you guys next week. Okay, right, so just last one, I will post a link on Spectrum on submission